welcome to the deep dive. Our goal here is, well, pretty simple. Cut through the noise, pull out the key insights, and basically give you a shortcut to understanding what really matters on a topic. And today, we're tackling something truly foundational. Nucleic acids, DNA and RNA. We all hear the blueprints of life, right? But what's really amazing, I think, is just the sheer elegance, how compactly they store and execute all the instructions for something as complex as us.
It is quite remarkable.
Yeah, I mean, you're talking about fitting miles of genetic code into a cell nucleus you can't even see. So our deep dive today is about uncovering those clever design principles, the stuff that maybe doesn't always get mentioned.
It really is staggering. And these molecules, they're absolutely central, found in every living cell, from the tiniest bacterium up to, well, us and even in viruses. So understanding them isn't just some biological detail. It's about grasping the very essence of how life organizes itself, maintains itself, passes itself on. It really is the ultimate instruction manual.
Okay, so let's unpack that instruction manual idea. We talk about nucleic acids, these huge biomolecules. But what's the sort of fundamental thing about how they work? Why are they so essential?
Well, the fascinating thing is, despite how powerful they are, they operate on a surprisingly simple principle. At their core, they carry and they transmit genetic information. That's the job. And when we say nucleic acids, we mainly mean two types: deoxyribonucleic acid, that's DNA, and ribonucleic acid, RNA.
Right. And the real genius, maybe, is how these incredibly complex molecules are built from, well, simple repeating units, nucleotides. It almost feels counterintuitive given how much information they actually hold.
It really does. And, most people probably remember the basics. A sugar, a phosphate group, and a nitrogenous base make up a nucleotide. But the real magic, the code itself, is in the sequence of those bases.
The order.
Exactly, the specific order. It's like having, say, just 4 letters in an alphabet, but the way you arrange them creates, well, endless biological stories. It dictates everything.
So DNA holds the master plan, the instructions. But a plan is just words until it's actually done, right? How does this blueprint turn into something tangible, like a protein our muscles need or an enzyme for digestion? What's the actual flow?
Yeah, that's where we see what biologists call the central dogma playing out. DNA, it stays safe inside the cell nucleus mostly. It's got all the instructions to build every protein your body might ever need, but it doesn't, go out to the construction site itself. That's where RNA comes in.
Okay, so RNA is the intermediary. Is it just like carrying the message, or is it more hands-on in the building process, like the difference between someone reading the blueprint and the site manager actually directing things?
Oh, it's definitely more like the site manager. RNA takes those instructions. Essentially, it copies or transcribes specific sections of the DNA into a portable format, a message. And that message is then used, translated to build the actual protein. So you have this flow, DNA to RNA through transcription, and then RNA to protein through translation.
Got it. It sounds like RNA and DNA, they're very closely related then, built from those same nucleotide monomers we talked about. But their jobs are so different. So I guess their structures must have some really key differences too. That must be critical.
Absolutely critical. Yeah, one of the biggest distinctions is their overall structure. DNA, famously, is that double helix.
Right, the twisted ladder.
Exactly. 2 strands elegantly twisted around each other. And those nitrogenous bases, they pair up in the middle, A with T, C with G, held by hydrogen bonds. This double-stranded structure, it makes DNA incredibly stable, robust, which is perfect for storing the genetic blueprint long-term. You want that to be secure.
Makes sense. Yeah. And RNA then? How's it different?
RNA is usually single-stranded, just one strand. And this makes it much more versatile, actually, and more temporary. It can fold up into all sorts of complex shapes to do different jobs, and it's designed to be broken down more easily once its task is done. Ideal for a messenger or a helper molecule that you only need for a short time. Plus, there's that other crucial difference you mentioned earlier in the bases themselves.
Right, the bases. So DNA has A, T, C, G, adenine, thymine, cytosine, guanine, and RNA.
RNA has adenine, cytosine, and guanine too, but instead of thymine, it has uracil, U.
So U replaces T. Is that just like a random swap, or is there a reason?
Oh, there's definitely a reason. It's not arbitrary at all. For one thing, uracil is actually energetically cheaper for the cell to make than thymine.
So for a molecule like RNA that's made and broken down constantly.
Exactly, it saves energy. But maybe more importantly, having thymine in DNA helps with error checking. See, cytosine can sometimes degrade and turn into uracil by accident. In DNA, if the repair machinery sees a uracil where a thymine should be, It knows immediately that's wrong, that's C degraded, and fixes it back to a C. If DNA used uracil normally, it would be harder to spot those CDU errors.
Ah, so using thymine helps protect the integrity of the permanent DNA code. That's clever.
It's a very neat system, subtle, but really important for long-term stability.
So if DNA is the master blueprint, the sort of central command, what are its main jobs? What does it actually do for us, day to day?
Well, it pretty much orchestrates almost everything about who you are and how your body functions. First off, it determines your inherited characteristics. Eye color, height potential, predisposition to certain things. That's all encoded in your DNA sequence.
What makes you you?
Exactly. Then, crucially, it directs protein synthesis, tells the cell which proteins to make, when, and how much. And proteins do almost everything, building tissues, carrying oxygen, fighting infections. It also controls enzyme production. Enzymes are just specialized proteins that speed up biochemical reactions. So by controlling enzymes...
It controls metabolism.
Precisely. It controls your overall metabolism, how your cells get energy, use nutrients, break down waste. It's directing all those fundamental chemical processes.
Wow. So it's not just a static list of instructions. It's actively managing pretty much everything, from how we look to how our cells are powered.
That's a good way to put it. It's the ultimate control center.
And does that level of control mean it has, like, backups or error checks? You mentioned the T versus U thing helping with repair. Can things still go wrong? Can one typo mess things up?
That's a great point. Yes, DNA has amazing proofreading systems and repair mechanisms. They catch most mistakes during replication. But they're not absolutely perfect. Typos mutations can happen and sometimes do slip through. And depending on where they occur, yes, they can have significant consequences, from minor changes to serious diseases. But its overall power comes from that stability, the double helix, and those very specific base pairing rules, A with T, C with G. That allows it to be copied faithfully, generation after generation, and transcribed accurately into RNA when needed.
Okay, so let's shift back to RNA then. We said it codes the intermediate steps for making proteins.
Yeah.
But you also mentioned it's not just one thing, right? It's more like a specialized team.
Exactly right. RNA isn't monolithic. It plays several very distinct specialized roles. Our sources really point out 3 key types involved in that protein building process, like an assembly line crew. First, you've got messenger RNA, mRNA.
A messenger, right?
Yeah. This is the molecule that carries the actual genetic code, the instructions for one specific protein from the DNA in the nucleus out to the cytoplasm, where the protein-making factories, the ribosomes, are. It's basically a direct copy, a transcript of a single gene.
Okay, mRNA carries the message. Then what?
Then you have ribosomal RNA, or rRNA. This type doesn't carry code itself. It's actually a major structural part of the ribosome.
Ah, so it helps build the factory machinery.
It is the factory machinery, in large part. rRNA molecules, along with some proteins, form the ribosome. And rRNA is what actually binds to the mRNA, holds it in the right place, and even helps catalyze the chemical reaction that links amino acids together to form the protein chain. It's the workbench and part of the tooling.
Wow, okay, so RNA is the reader and the assembly station. What's the third type?
The third key player is transfer RNA, or tRNA.
Transfer, so it moves something.
Precisely. Its job is to ferry the correct amino acids, the building blocks of proteins, over to the ribosome. Think of it like a delivery truck. Each tRNA molecule is specialized. It has a little sequence on one end that recognizes a specific 3-letter code word called a codon on the mRNA.
Okay.
And on its other end, it carries the one specific amino acid that corresponds to that codon. So as the ribosome reads along the mRNA codons, the matching tRNAs arrive, deliver their amino acid, and it gets added to the growing protein chain.
Since you really can picture it like that assembly line. The mRNA is the blueprint scrolling by. The rRNA is the workbench and the main assembler, moving along the blueprint and holding everything.
Right.
And the tRNAs are these little trucks constantly bringing exactly the right part, the right amino acid at exactly the right time, matching the code on the blueprint.
That's a perfect analogy.
It's just an incredible dance of molecules, a symphony, really, happening at unbelievable speed in every single one of your cells all the time.
It is truly mind-boggling when you think about the scale and the precision.
Yeah.
Billions upon billions of these operations happening right now as we speak.
So we've covered quite a bit today. We've really gone deep into this amazing world of nucleic acids, DNA, the master blueprint, and RNA, the versatile construction crew. They really are fundamental to, well, everything alive.
Absolutely. It's hard to overstate their importance, the complexity, but also the elegance of how they work. It really does capture the essence of life's processes, storing information, executing commands with such precision. And what's still so fascinating, I think, is how this seemingly simple system, just a few building blocks, specific pairing rules, can generate the incredible diversity and complexity we see in all living things, including you listening right now. It kind of leaves you wondering, doesn't it? What other fundamental blueprints or systems might still be out there waiting for us to uncover that explain even more about how life works? Maybe things beyond the genetic code as we currently understand it. Something to think about.